Greetings and welcome to the latest installment of the SLAC Public Lectures. This year we're celebrating the 60th anniversary of SLAC, and many of the themes of this anniversary have to do with new scientific activities in which SLAC is pioneering. And so many of the public lectures this year will deal with those themes, and this is certainly one of them. We have this amazing new facility called Cryo-Em, which will be explained in this lecture, which produces amazing images of biological structure. And we're going to hear today a lecture that takes advantage of that facility to tell some amazing things about the biology of viruses. The speaker is Rachael Kretsch. She's a graduate student in biophysics here at Stanford, a student of Professor Riju Das, and one of those people exploring now new details of viral RNA structure that only become apparent with this technique. She has an interesting history. She comes originally from the Netherlands. She studied at Harvey Mudd. She's wandered around the world a little before landing at Stanford. And now she's engaged in this very interesting activity, which she's about to tell you about. Let me just say one more thing before we begin. We will have a question and answer session at the end of this lecture. You'll see at the bottom of your Zoom screen a Q&A box. And so please open that up. If you have a question, type in your question, and we'll ask all those questions of Rachael and her colleagues who are here as panelists after the end of the lecture. So with no further ado, I introduce Rachael Kretsch, and she's going to tell you about revolutionary 3D views of viral RNA. So please.
Great. Thank you, Michael. So as Michael said, I'm Rachael, and I'm going to be telling you guys today about RNAs, their structure, and how we study it with Cryogenic Electron Microscopy, or as how we call it, Cryo-Em. So let's begin with just a brief introduction of who I am, some of which Michael has already gone over. So I grew up in the Netherlands in a beautiful city called The Hague. And there, some of my exposure to science was really with this organization called OPCW, which works on chemical weapon issues. So from an early age, I was really engaged not only in science, but also how it can impact society. Then I headed to Harvey Mudd, which is a great little school in Southern California. There I studied the 3D structure of molecules. So there's just an example at the bottom of the slide. And I also fell in love with the mountains around here and hiking. And one of the reasons I'm back in California now for my PhD. I took a year to then dive into my interest in science and society, and specifically science and security in London. You can see the beautiful Christmas decorations around where I lived. And also got the opportunity to work with wonderful researchers at two centers, one in Monterey and one in Los Alamos. And that's how I ended up here. So now I'm at Stanford, and I'm part of the biophysics program. And I'm also a fellow at BioX. So what I do is truly interdisciplinary. So I study biological questions, but I do so using knowledge from chemistry, physics, and computation. And I do this in two labs. So first, Riju Das's lab, the Das lab, which are experts on this molecule called RNA and what its shape or structure is. And then the Chu lab, which are experts on the electron microscope that allow us to see RNA. And because we need these very powerful microscopes to see RNA, I do all my imaging here at SLAC. So at the center called S2C2, that has a few of these microscopes, and I'm lucky enough to get time on them to see my RNAs. And I also just want to highlight that no scientist can do their work alone. We're all surrounded by a massive community of other scientists. Here's just a small selection of some people in my community. And I want to highlight the following people that contributed or led the work that I'll be talking about today. So in blue, we have Kaiming, Xiaoming, and Callie. And they were the three that led the original effort to show that Cryo-Em, this technique I'm going to be talking about, can be used to solve the shape of a variety of different RNAs. And then in pink, we have Vanya, Kaiming, and Rachael, who led the effort to study the RNA that I'm going to be talking about as an example today, that is from the SARS-CoV-2 virus, the virus that causes the current COVID-19 pandemic. All right, so I like to start my lectures with the takeaways, and hopefully by the end, I'll convince you of some of these takeaways. So the first is that RNA viruses are a large challenge to human health, but structural biology offers us clues on how they work, and thus how we may think about stopping them. And then we have decades of technological development and basic science that enable our current response to these viruses. RNA is a key component of these RNA viruses, but its structure has remained a mystery for much of history. And then Cryo-Em is finally enabling us to actually look at these RNAs. So we're going to split this into 5 parts. The first is, what is structural biology? Then we're going to learn about what RNA viruses are. We're going to learn that there's 4 levels of RNA structure, and we're going to go through the example. And then we're going to show how we study RNA 3D structure using the Cryo-Em. And then finally, we're going to ask what these structures can actually tell us about the virus. So let's start with what structural biology is. So if you Google structural biology, you're going to end up with a bunch of images that look something like this. And quite frankly, I wouldn't know how to interpret or what a lot of these images are. But the basics to structural biology is we care about the shape of these molecules. So for example, we have 3 rings. And the ring is probably important for what these molecules do. And we have other shapes like spheres or diamonds. So to simplify it, let's look at an analogy. And in this analogy, we're going to have a character that's going to start at the starting line here. And the question we're asking is whether this character can make it through this tube to reach the other side in our 3D scene. So if I give you the first level of structure, it's just the list of building blocks. So we know the character has a red hat, brown shoes, a black mustache, but that's all we know. And that's insufficient to tell us whether it can fit in the tube. Then we go to the second level of structure, which is how the parts are connected. So we know the shoes are at the bottom, they're connected to the overalls, and then the hat's on top. But we still don't know the exact dimensions or shape that would allow us to ask whether it can fit through the tube. We need the 3D structure for that. And once we have the 3D structure, we can fit him in the scene. And we see he's slightly too tall to fit into the tube. But this is because we've missed the 4th level of structure. And that's the fact that these shapes can change and move. So in this case, our character is able to lower his hands and then lay down in order to get through the tube, right? Now, if that's what he wants to do, so he wants to make it to the other side, is there something we can do to stop him from being able to get through the tube? And the answer is yes. We can give him a bunch of coins. Now he's busy holding his coins and can no longer lay down and make it through the tube. So we're also interested in how our character interacts with other things like the coins. So how does this relate to biology? Well, in biology, the scene is the cell. And the characters in the scene are what we call macromolecules, which just means big molecules. Now remember, it's in 3D, so you can see down here, we have a 3D shape that's kind of like a mouth. And this is actually the ribosome that we'll be talking about a little bit today. So biology happens when macromolecules interact in a cell. And we have 2 questions we want to ask about them. How do they work? So in the case of Mario, he put his hands down and laid down to make it through the tube. And then how can we stop it working? So in the case of Mario, we gave it a bunch of coins. But in the case of viral biology, it's often through drugs. All right, so what macromolecules are we talking about? Well, today we're really focusing on RNA, which I pictured here. And RNA is a close cousin of DNA that you may have heard of. DNA is what humans store their genetic information in. And RNA in humans is just a direct copy of the DNA. So you can see we just directly copy over the colors from DNA to RNA. RNA is a sequence of 4 bases. So we have A, C, U, and G. And the full name is ribonucleic acid. So I'll probably will never say that again in this lecture. We tend to call it RNA. And one of the main functions of RNA is to make proteins. So RNA gets what we call translated to proteins. Now, we see a problem here. RNA is made out of 4 bases, right? A, C, U, G. But proteins have 20 building blocks, or what we call amino acids. So how do we do this? Well, it reads it 3 at a time. So we have 3 bases of RNA that make up 1 amino acid. And then the next 3 make up a different amino acid. And then we continue on reading the RNA 3 at a time and translating it to 1 amino acid. And we continue onwards. Now, in reality, our proteins are larger than the small protein I pictured previously. So for example, the spike protein of the SARS-CoV-2 that you may have heard about is 1,000 amino acids. So that means 3,000 bases of RNA go into translating into that protein. And this simple model of how DNA and RNA and protein are related is just that simple. So RNA is not just a messenger, and hopefully today you'll see 1 or more examples of that. And RNA has shape too. So I've kind of just drawn it as a string, but there's plenty of different RNAs that have a 3D shape just like that protein does. So let's watch translation happen, because that's what we're going to be focusing on today. So this is a 3D video. It's not real life, it's just a simulation. We have the RNA in yellow here, looking like a string. We have a protein in red up top, and then this blue is the ribosome. That's the machine that's doing the translating. So we can watch the ribosome pull the RNA through it, making the protein up top. And what it's doing is reading 3 bases and putting 1 amino acid on top. Okay, notice all these 3D structures moving around and fitting in nicely together. And the protein comes out the other end and folds up. Now, it would be great if we could actually see this in real life, but unfortunately, all of this is much too small for us just to see. So we're going to zoom in into the size of RNA, starting at size 12 font and a grain of rice. We are going to be zooming in past a grain of salt, past a skin cell. So that's your human skin cells. We now are going to start seeing viruses like HIV. There's the ribosome, that was that big machine. And then here we have RNA. And if we zoom back out, we can see we're getting bigger and bigger. The RNA we can no longer see at this point. We have yeast, which makes your bread. And we zoom all the way out back to a grain of salt and coffee bean. So biology is very, very small. We can't simply see it. We can't even use one of these microscopes. So we use an electron microscope. And what that means is it magnifies it 100,000 times. To put that into perspective, a human would now be 18,000 football pitches long. But our RNA started so small, at this type of magnification, it is now around 1 millimeter, or the size of a grain of rice. So it's now visible, but still pretty small. Now there's a second problem to look at structural biology, and it's the fact that this cell is incredibly crowded. So here's an artist's rendition of what a cell may look like. And you can see I've labeled here the ribosome, that was that big machine in the video, and the RNA that's getting translated in pink. And you can see lots of ribosomes translating different RNAs. But there's also a lot of other stuff in this cell, right? It's a very crowded environment. And you can imagine if I was to take a picture of this, it would be very hard to point out which was the RNA and what was other things, right? So we don't actually image the cell. we just image RNA alone in salty water. So we don't have to deal with picking it out and differentiating it from everything else in the cell. So I'm gonna release a secret that structural biology is not gonna solve the world's problems. It is more of a tool to give us insights into solving the problems. So I liken it to a textbook. So a textbook does not solve your problems, but you still consult the textbook when you want to solve a problem because it gives the necessary background on how to solve the problem. Structures are very similar to biology. They will not tell you how the macromolecule works or what drug will stop it working, but you still solve a structure when you want to understand the biology because it forms your hypotheses on how it may function and what drug may affect it. So they inform how macromolecules may work and how we may stop them working, but it's not the end of our work. We still have work afterwards. All right, so let's learn what RNA viruses are. And the first thing to realize is how important they are. So here's a list from the World Health Organization of what they deem the diseases of most pandemic and epidemic concern. And I'm just going to highlight which of these are RNA viruses. It's the vast majority of them. So you can see some viruses like influenza that causes flu, or even the coronavirus that we're currently battling. So what are these viruses? Well, here's a picture, an artistic picture of the coronavirus. And in yellow and green, we have the mucus of our lungs. So that's the environment the coronavirus is floating around in. In pink, in the sphere, we have the coronavirus spike protein that you may have heard of. And then packaged away in the middle is the coronavirus RNA. And that's what we're really interested in studying. And here's one reason why. When the coronavirus in pink here infects the human cell, so you have the human cell in blue, what does it actually inject? Well, it's the RNA. So the RNA of the coronavirus is what enters our cells. It starts to get translated to make viral proteins and also gets copied to make more and more copies of the virus. So we're going to refine our DNA to RNA to protein. For RNA viruses, it really truly is just RNA to protein, right? So we're getting viral RNA, and then we're translating that to make viral proteins. Okay, how do we study the RNA? So we have 4 levels of structure. And I already went through with the analogy of our character here, the 4 levels of structure. So we have the list of building blocks. Then we have how all these building blocks are connected. Then we have the actual 3D shape. And finally, we have how it moves or interacts with other things. And in RNA, we have very similar 4 levels of structure. And we have different types of experiments that can give us information at the first level, or the second, or the third, or the 4th level. And as we go up in levels of structure, we learn more and more about our RNA and how it works and how we can stop it working. So let's start at the first level, which is simply the sequence. So you already know there's 4 building blocks, G, U, A, and C. And the sequence is simply the order that these building blocks are in. So we want to determine the primary structure, or the sequence, of the RNA of coronavirus. And this was done incredibly quickly, probably before many of you had known the existence of COVID-19. So in January 10th, 2020, the first full sequence of the coronavirus RNA was published. And how was it that we could get this so quickly? Well, it really was technological advancements that allowed us to do this. So this is a graph. We start in 2001 down here, and we go to present day. And here we have the cost of sequencing. So the white line is halving the cost every year. So that's already a great trend. But you can see we did much better than that. And that was because new technology came out called next generation sequencing. And what that allowed is now the cost of sequencing one SARS-CoV-2 genome is only around $34 by some estimates. So now we can sequence, genomes from around the world and from different patients. And I just want to point out on this timeline that we had an outbreak of a cousin of SARS-CoV-2, SARS-CoV-1, back here. It was only roughly 15 years ago, but the cost to just sequence this virus would have been much more expensive, and it would have been much slower. All right, so here's the primary sequence. It's simply a list of ACs, Gs, or Us, and there's 30,000 of them roughly. Now, just reading a bunch of AC, Gs, and Us is not going to tell us much information, but we can compare it to other viruses to identify closest relatives. So that's how we knew it was a coronavirus, for example. We can also track changes in this sequence, which tells us new variants that you may have heard of. And what's of interest today is we can actually identify what proteins are being made, because we know through translation, there's a direct mapping from the RNA sequence to the protein sequence. And that was an interest for the spike protein. So for the spike protein, the ribosome will bind here in the RNA sequence, and it will translate this part of the RNA to make the spike protein. Here's the spike protein over there. It's what our cells see, and that's why it's so important, because it's what our human defense or the immune system sees and tells us that we're being infected by the virus. All right, so we wanted to know the shape of the spike protein, and due to the advances in Cryo-Em, this was solved in little over a month. That is incredibly fast, and we were very impressed. Now, one thing to note, right, is this is a protein. The field is much more advanced to proteins. I work on RNA. We're not quite able to solve these structures within a month yet, but the fact they were able to do this for a protein gives us hope that maybe years down the line, we'll be able to do this with RNA as well. So I said, Cryo-Em was an important factor here. Let's go through what Cryo-Em is. So here we have a schematic of Cryo-Em. We start in the middle here, where our protein or RNA is frozen in ice, okay? And you notice some of them are up, some of them sideways, some of them downwards. So we have multiple copies of our protein or RNA, and they're all in different orientations. And then we have an electron beam that comes through and hits the protein or RNA and makes an image on the other side. And these images, some of them are upward facing, sideways facing, bottom facing, right? And then we take thousands or millions of these images, we organize them by their orientation. So we have sideways facing, upwards facing, etc. And we use a computer algorithm to reconstruct the 3D shape from these images. So I'm going to focus on how we actually get this sample. So in my case, I just have RNA in salty water, and I apply it to this grid here. And I try to get the thinnest layer possible, and then I dip it into liquid ethane, which is extremely cold, negative 196 degrees, to freeze it very quickly. So now I have a very thin layer of ice, and all my RNAs are frozen in time. And I keep it at this temperature, so that's why it's called cryo, which means cold, as I image it. And you can see here, I have now a nice thin layer that my electrons are going to pass through. So let's look at that in a little more detail in this video. So we have a grid, it's made out of copper, and it's only roughly 1/10 of an inch in diameter. So you can imagine it's very, very small, and we handle it very, very carefully. We're going to zoom in into one of these squares. And these squares here are covered in a layer of carbon. And then we have these holes, which is where we're going to do our imaging. So in these holes, it's simply a thin layer of ice with my RNA or protein in it. And we can zoom in and look at this view. You see here, in this case, it's ribosomes actually, so that machine we were talking about earlier. And they're all in different orientations, right? Some upwards, some downwards, but they're frozen in time. All right, let's look what it looks like to image. So we're looking from the top now at our hole, and then we take an image. So the electrons go through and make an image on the other side, In reality, it's much noisier than this. This would be a perfect scenario. And then we extract the images and we sort them by the orientation. So we have a group that's side looking, a group that's from the top, and a group that we're looking from the bottom. And then we use a computer algorithm with all of these images to reconstruct a 3D volume here. And you notice as we put more and more images in, we're able to get more and more detailed view of what the shape is. So you can start to see much more details. So that's the basics of Cryo-Em. And just by knowing the RNA sequence, they were able to put a bunch of RNA proteins on these tiny little grids and image them with Cryo-Em. And these are just some of the shapes they were able to solve. When I say they, I mean the scientific community, lots of labs, lots of collaborations. All right, and this, structure is not the end. So you would then use this shape to identify how these viral proteins work and how we can actually stop them working. All right, so I want to focus in on this translation. So we have a group of orange proteins here where the ribosome binds at the beginning of the virus and starts translating to make these 11 orange proteins. And then it hits a stop signal and it stops translating. We also have a group of blue proteins here, more in the middle of the virus. And I wanted you to notice something very interesting. There's an overlap between the orange and the blue proteins. So what that means is that the same RNA sequence is being translated into an orange protein, but also part of a blue protein. So how does that work? How does the same RNA sequence give us two different proteins? Well, let's look at the blue mechanism. So the ribosome starts binding here still, and it translates a bunch of orange proteins, just like the orange. But when it reaches this pink box, it slips. And this slip causes it no longer to make orange proteins, but instead start making blue proteins. Okay, that's great, but why does any of this matter? So let's zoom in on this region right here where this slip is happening. Now this slip in coronavirus happens around a quarter of the time. So a quarter of the time it just reads like normal, no slip, 3/4 of the time. Now what will happen if I add a drug that stops the slipping? So now I'm going to get less of the blue. What we like to measure is viral replication, right? Because every virus, their goal is to copy itself and make more virus. So we're going to measure how well it can copy itself. So when we have no drug, it copies itself pretty well. But as we add this drug that stops the slipping, the virus also fails to replicate, right? So we're actually harming the virus by stopping this slipping mechanism. So it's important. We want to study how it's slipping so we can try to prevent it from slipping. So let's zoom in again on this region, right, where the slipping is happening. And we're actually going to look at the RNA sequence. So here's the RNA sequence. Again, this RNA is getting translated by the ribosome, right? So let's just remind ourselves what translation is. We have the ribosome, this big machine in blue. The RNA is being pulled through the ribosome, and it's reading three bases at a time to add one amino acid to our protein that's getting formed up top here. All right, so let's do that. Let's translate this part of the RNA sequence. So UUU gets translated into the amino acid F, UUA to L, the next 3 to an N, then a G, then an F. So that's how the ribosome reads normally, 3 at a time. Now what happens when we have slipping? So it starts normally, we have FLN, but this is when the slip happens. And what the slip means is instead of reading GGG, it slips back one and reads CGG, which makes R instead of G. So we start making different proteins. And then it continues reading GUU, which is V. So now we're making completely different proteins just by slipping back one base. As an analogy, let's look at some English words. So we have a list of letters here, and I read out the words comic Roman goose. All right, what happens if I put a slip in there? So I'm starting to read normally comic, then I put a slip, so I move back and I read micro mango, comic micro mango, right? Very different meanings, very different words from the same list of letters because I slipped. And we call this a frame shift because we're shifting the frame that we're reading or translating in. And the RNA here and further on is what we're interested in studying because we think its shape is causing this slip to happen. So this is what we call the frameshift stimulation element, or as you probably noticed by now, we like using acronyms, so we're just going to call it FSC for short. And the FSC is this region of the RNA, the region where the slip is happening. So what are we curious about? Well, we want to know the shape of the FSC. And why we want to know it is because we want to know why sometimes the ribosome reads normally and why sometimes it slips, with the overall goal of informing us how it inhibits, how it frame shifts, and then how we could potentially inhibit the frame shifting and thus inhibit the virus. So what's the primary structure? Well, that we had back in January 2020, luckily enough. But frankly, we don't really know much just based on the primary structure, right? It's just a list of U's, A's, C's, and G's. I'm actually going to give you a sneak preview and color it by what's near each other in 3D space. So you see, for example, the blue, we have these regions that are quite different in sequence, but far from each other in 3D space. So let's learn more. Let's go to the second level of structure, how all these things are connected. So for RNA, it's quite basic. So we have A's, C's, U's, and G's, and they just pair up. So U's and A's like pairing together, and C's and G's like pairing together. So if we take this sequence, we can pair up this G and this C, and then we can pair up that G and C, and we can keep going, pairing up that U and A, this A and U, this A and U, this C and G, and you can see we have a nice ladder of pairs now. But then we get to a G and an A. Those don't like pairing up, which is absolutely fine because RNA doesn't mind being unpaired. It prefers being paired, but it's fine with being unpaired. So now we have what we call a stem, which is this ladder of base pairs, and then the loop, which is this unpaired region. So let's look at the frameshift stimulation element, or the FSC. So we're going to start where the ribosome is bound. So it's here. And we have an unpaired region. Okay, so it doesn't have any pairs. And remember this, the ribosome is pulling the RNA through it to translate. And a quarter of the time, this is where it slips. So what is it trying to pull out first? Well, the ribosome is first going to try to pull this green stem here, which has that blue loop. Then we have an orange stem with that black loop, followed by a pink region that's not paired. And then we have the end of the FSC. Now, did you notice how I colored these both blue? Well, it's because they can actually pair together. So you see we have G and C, G and C, G and C, C, G, U, A. Now I said RNA is fine being unpaired, but it prefers to be paired. So what happens is it pairs up. This loop is no longer a loop, it becomes a stem. And when this happens, when a loop becomes a stem, we call it knot-like. So the FSC is kind of like a knot. Now, why does this matter? Well, the ribosome is translating, pulling through the RNA. And you can imagine if you're trying to pull something that's not quite a knot, but knot-like, you get more and more resistance. So this resistance may cause it to pause and thus slip. So how could we think about stopping it from slipping? Well, what if we get rid of the knot, right? So what if we get rid of those base pairs? How would we perhaps do that? Well, what if we came up with a drug that really liked binding this blue and pink region here? And that would leave this to be a loop and is much easier to pull. Now this drug is completely hypothetical, but you can imagine it kind of like the coins for Mario, right? So this region likes binding that drug so much that this drug will bind and leave this to be easy to pull, and therefore we get no more pausing and no more slipping. All right, well, let's learn a little bit more by going into the 3D structure. So the basics of 3D structure is just on the stem. Now, if you noticed before, I said it was called a stem or a helix, and that's because of what it looks like in 3D. So we have a ladder of pairs, right? But in 3D, that ladder is not a straight ladder. It's like a winding staircase, or a helix. You can see the pairs and they're going around in a helix. And the loop is simply this region up top. Now, we don't actually see each of the bases when we look at our Cryo-Em data. So we actually just see the shape when we're looking at Cryo-Em. So this is the shape we see. You can still see kind of, it looks like a helix, right? A little staircase going around. But one of the challenges when we're working with Cryo-Em is the question of how do we fit these bases in this general shape, right? So we use Cryo-Em to get the shape, but then we still have the problem of how do these bases fit in that shape? All right, so let's focus on how we get the shape first of all. And that's really done through Cryo-Em right now. At least that's what we do at SLAC. And why is this so revolutionary? Well, I think it becomes obvious when we look at the past of structural biology. So structural biology has managed to solve 160,000 protein structures, right? Huge amount of protein structures. How good are we with RNA? Well, we're around 100 times less structures solved for RNA, right? And this is due to many challenges with traditional techniques. But it is our hope that Cryo-Em can help us solve these RNA structures, unlike other past techniques. So here's a review of what Cryo-Em is. We have our RNA, in this case, suspended in ice here. So remember, the RNA is sometimes pointed up, sometimes down to the side. We have electrons that come through our sample to make a 2D image on the other side. And some of the views are top views, side views, bottom views. And then we take all of these images, in our case, often millions of images, and we ask a computer algorithm to reconstruct the 3D volume from these 2D images. And why do we think it's so promising to use Cryo-Em technology? Well, I'm going to give two main reasons. First of all, it's in water, right? And why is that so special? Well, before we used to have to make crystals of our RNA. And all a crystal is pretty much a densely packed RNA. So we have all of our RNAs packed densely into this crystal. And RNA just doesn't really like being packed that way. So it took a lot of effort and time to get good crystals. The second reason for water is it's closer to the cell-like environment than a crystal would be. I say closer, right? Because it's just floating around in water. It's not the crowded environment of the cell. And hopefully maybe years down the line, we'll be able to actually image in the cell. And the second reason is it's faster. So I can get more data quicker for different RNAs. So let's look at some structures that were solved. So this is work done in 2020 by Callie Xiaoming and Kai Ming that led it. And you can see they solved a variety of RNAs. So they really show that Cryo-Em was capable of solving the shape of many, many RNAs. So in gray is the shape. So that's the RNA, the Cryo-Em data is in gray. And then in colored is what we think the base positions are. So where do we think these bases go in this shape that we got from Cryo-Em? So you can see we have many different types of shapes. This one, for example, was designed to be clothespin-like shaped. And this was actually the smallest shape that was solved by Cryo-Em at that time. And why do I point that out? Well, size is a huge limiting factor for Cryo-Em. So remember, as we were zooming in, how small RNA is? Well, even at the 100,000 times magnification that our microscopes allow us to see at, our RNAs still look like tiny dots. So this was the smallest RNA previously solved. And the FSC, when we're interested in solving, is 5/7 the size of that. So it's even smaller. So this was going to be a huge challenge. But the team at the DOS lab and the CHU labs were up for the challenge. I want to show you guys the raw data. So this is what the image out of the microscope looks like. And we have circled the RNAs. So they're really, tiny dots. You can see we don't have much detail when we zoom in. But if we collect millions of these images and average them, we can start getting more detailed pictures. So you can start seeing the shape in these 2D images. And with enough data, we can start to reconstruct the 3D shape. And here it is. This is the 3D shape of the FSC. So this is what the ribosome's trying to pull at. And you can see we pointed out a hole, right? We're not really sure if what that hole is, but I really want to focus in on this region right here. And if we turn ourselves 90 degrees, we're looking at it here. And can you guys see how that's kind of that staircase or that helical shape? So this is a clear stem. So we just have a stem and then a loop at the end here. And if we turn it 90 degrees again, you can still see that helical-like shape. So let's try to model in our bases now. Where do the bases go in this shape? So we have this shape, but we also have the secondary structure we went through before, right? So we have how many stems we have. And you notice how we have one clear stem jetting out here. Well, we also have a stem jetting out here, right? So that's in fact our orange stem over here. And then we use a bunch of computational modeling techniques to fit in our RNA bases. And here's how they fit in. So we have our orange stem jetting out here. And then we have the green and the blue stem that cause that knot-like structure just stacked on top of each other here. And then we have the pink connecting these two. So, okay, great shape. What does it actually mean, right? Structures don't tell us enough. We need to study them more. So let's look at a video to see this shape. So you can see as we turn it around, you can see the shape. Now we're going to, this is where the ribosome's pooling, and it first pools this green helix. Then we have this orange helix. Then we have the pink and the blue. And now you see how the bases don't fit perfectly into the shape. So we're a little uncertain in what the shape is. So that's just showing you that we have 10 different possibilities of how the bases may fit into this Cryo-Em shape. And now we're going to remove the part that the Cryo-Em is pulling on next and draw it back in. So did you notice how when we drew it back in, it was right between the pink and the green, or threaded through the pink and the green parts of the RNA? We're going to look at that in a little more detail now. So I'm going to color the part that the ribosome is going to pull next in red. So the ribosome is trying to pull on that red strand so it can translate it. All right, let's zoom in to see this more clearly. So the red is what the ribosome is going to translate next. So it's trying to pull it into the ribosome. And you see how it has to go in between the green and the pink. All right, let's watch the video one more time. So we're going to remove the part that the ribosome is pulling on next. And then we're going to model it back in, and you see how it threaded through the pink and the green parts of the RNA. Just one more time, we remove it, Now we can see where it goes. It's going to thread directly through the pink and the green. Okay, great. It's threaded through the pink and the green strings. Why does that matter? Well, let's think about how the other option, the unthreaded option. So if we zoom in, we see the threaded again. It has to go through the pink and the green parts of the RNA. The unthreaded, it just goes around them, right? And why will that matter? Well, if it has to go through these RNAs, it's going to get more resistance, right, as it's pulling it through. So maybe that causes it to stop and slip. Whereas in this case, it has less resistance because it doesn't have to go through or thread through. So maybe that lets it just continue normally and read normally. Now, one thing I want to note here is we did not see this. So this is just a hypothetical structure. This is the structure we saw, right? Okay, so we have a hypothesis of how it may work. Could we design a drug to stop it working? Well, what if I put a drug here in this hole that kind of acted like a glue to keep it threaded? So to make that resistance when we're pulling even harder. And that would cause it to slip even more, right? Because it's even harder to pull. So that may be a drug that could work. Again, just a hypothetical drug. All right, so we have that information and I'm telling you a lot of ideas about how we think the ribosome works with it. But remember how on the grid there was just RNA and salty water, right? There was no ribosome. So we really want to know how the ribosome interacts with the FSC. So what that means is instead of just having RNA and salty water, we need to add ribosomes into the mix. And luckily, teams from Zurich and the UK did this study and published in 2021 in Science using Cryo-Em with the mixture of ribosomes and this part of the RNA to reveal this interaction. And this is what the interaction looks like. So in gray here, we have ribosome. Remember, the ribosome is this huge machine. So it goes off of this picture as well. And it's currently translating the RNA. So this is the coronavirus RNA. It's currently translating. But we captured it when it's paused. So it's paused in translating right now. And this is the FSC. So it pauses right on where it's supposed to slip, right? And let's compare our structures. So we have the orange stem here. The orange stem also juts out when the FSC is interacting with the ribosome. We also have the green and blue stacked on top of each other, very similar. And then finally, we have this pink region, which connects the orange and the blue. Okay? So we have very similar structures. In fact, this structure is also not like, and it's also a threaded FSC. And I'm just going to show you that threading. So the ribosome is pulling here, and it's actually actively pulling in this case, right? Because the ribosome is there. And we know the ribosome is paused, right? Because the ribosome is physically in our images now. So let's just color the string red again, the one that the ribosome is trying to pull on next. And if we zoom in, we can see it's threaded through again. So we have the green and we have the pink, and the red is threaded through and the ribosome is trying to pull at that structure. All right, so we go back to our threaded and unthreaded examples. So now we have two images of the FSC, one from art groups of just the FSC, and then one from groups in Zurich and the UK of the FSC on a paused ribosome. And both of them are this not like threaded structure, right? So we have more support for thinking that this threading or not like structure causes pausing and thus slipping. Now, we don't have further support for this hypothesis, but we haven't ruled it out either. So how does this all pan out with the drug hypothesized. So I said, what if we bind a drug here that acts as a glue to make it even harder to pull the red through the ribosome? Well, if we look at that position in the structure on the ribosome, so I've just rotated the image, we can see that hole still exists. So we still have a hole that a drug may fit in. So it's a potential target, and that's what we like having structures for, to inform these potential targets. All right, let's watch a concluding video to see this in action. So we have the coronavirus RNA here in orange. That's where the ribosome slips a quarter of the time. And then this is the knot, the RNA structure that we're really interested in there. And in this case, it did not pause. It could just read through and it reaches its stop and dissociates and has made its viral proteins. But one RNA gets translated many different times. So now the knot is going to reform and another ribosome is going to come along making more viral proteins. And this time, the ribosome is going to pause and slip back. So this will happen a quarter of the time, roughly. And now it's reading completely different proteins. So it continues on reading these blue proteins until it reaches its own stop signal down the line. And that's how we think perhaps the RNA structure that's not like threaded structure will cause the ribosome to slip sometimes by making it harder for it to pull. So hopefully today you've learned what structural biology is and how we think about it. You've learned what RNA viruses are. You've learned that there's 4 levels of RNA structure and each give more and more information. You've learned how we use these cryo-electron microscopes to study 3D structure. And then finally, you've learned how we think about what these structures could tell us about viruses. And our key takeaways, well, RNA viruses are still a huge challenge to human health, but we hope structural biology will offer clues on, first of all, how they work, and then secondly, how we can stop them working. And decades of technological development and basic science have enabled our current response. And we hope our basic science work on RNA structure will enable future responses to disease. So RNA is the key component or a key component in RNA viruses, but its structure has remained hard to see. So we're working on developing the basic science through Cryo-Em to enable these revolutionary views of RNA in the hopes that we can learn about viral RNAs and how to stop them working. So looking to the future, we have many goals, but I'll just list 4 here. The first is to get higher resolution, where all that means is more detailed shapes, which means we're going to be more confident in where all the bases are. Okay, so if we have more detailed shapes, we can better fit in where all the bases should go. The second is the fact that RNA can change shape, right? So when I was talking about, oh, maybe there's an unthreaded RNA, well, that's a different shape. So we hope in the future we can see different shapes. Third, we hope to see how RNA interacts with proteins and drugs. And that could also be an avenue. And then in general, we just want more structures and how they're linked to how RNA works. And this is beyond SARS-CoV-2 to other viruses, as well as beyond viruses, because RNA is in all living things. All right, so here's the community, and special highlight to Xiaoming, Kaiming, Vanya, Callie, and Rachel, who led some of the work I was talking about today. And then here's a list of people in the community. So we have the Chu Lab and the DOS Lab, which has had many mentors for me learning how to study RNA and how to use Cryo-Em. We have past Chu and DOS Lab members, as well as other collaborators that have contributed to the work of RNA Cryo-Em. We have the great team at SLAC in mostly the communications department that organizes these wonderful lectures so that we can share our science to the community broadly. And then finally, we have our 2 panelists who we're about to hear from, Jonesy and Miri, who are great RNA scientists, and you guys are very lucky to be able to hear about their science, and that's what we're going to move to next. So I'm going to stop sharing my screen, and we are going to start the panel discussion.
Okay, well, Rachel, thank you very much. There's a lot of unexpected detail in this lecture. I must say I've learned a lot about this RNA biology by interacting with you. So I hope other people find it equally fascinating. You've introduced the two panelists, Miri and Jonesy. Jonesy, could you turn on your video? You're there somewhere. Actually, physically, you're in Munich, so it's like the middle of the night. But nevertheless, thank you very much for being here. And let's now start with the questions. So Stephen would like to know, is there a problem that the salt solution that you shake up the RNA in will change its shape? And I guess also Rachel talked about the crystallization. Is one better than the other? What's the deal here?
Yeah, so I'll try to answer that, and Miri or Jonesy, feel free to come in after. So first, salt. Salt is very important for RNA structure. So actually many RNAs will not form their structure if there's not salt around. So varying the salt concentration is actually something very interesting and could potentially change the shape or the range of shapes that the RNA can form. As for kind of crystallization versus using this frozen sample, all techniques have their artifacts, right? So we're not imaging things perfectly in the natural living cell, right? So I wouldn't say one is better than the other. They could give different results, but it doesn't mean one is better than the other. Quite frankly, it's just easier to get the RNA on the grid than to crystallize it.
Okay. Are you worried that this electron beam that you're using to make the image is going to destroy or deform the RNAs? Do you have control over that?
It does. It definitely destroys the RNA. So that's a factor we have to be very careful about. So we call this what we call radiation damage. So electrons are pretty powerful things, especially when you're dealing with such small scales. So if we put too much electron dose, we will damage our RNA completely. So we try to limit the amount of electrons we expose our sample to. And that's also why you saw so much noise in those raw images, right? They weren't clear at all. If we had exposed it to more electrons and there was no damage, we'd see much clearer images. But we simply can't do that without damaging the RNA.
Maybe you need more sensitive detectors. Of course, that's something also that we do here.
Yeah. Detectors, I'd say a lot of the current improvements that we've seen of Cryo-Em over, the past 10 years or so have been because of improvements in detectors. So now we're able to more sensitively detect individual electrons as they pass through our sample. And certainly improvements that allow us to do that faster and with less electrons will help in that. So there's many people working on that. I don't work on that personally, but I can only hope they'll give us much better detectors in the future.
Okay, Fred would like to know, do the RNAs get oriented in this setup? He interprets what you said as saying that you make 2D images, which then are combined to make a 3D image. So do you actually see the various turns or rotations of the molecule? Are they specific orientations that you see? And maybe you could say a little more about how you put the picture together to get a real 3D.
Okay, so I'll start with, what are these orientations? Do I get to decide them? And the answer is no. I don't get to decide how the molecules all fall. So you can think about it as, my RNAs are all in water and they're just all tumbling around randomly. And then I very quickly put into this very cold liquid ethane. So they all just freeze in place, however they were tumbling, right? So they're all kind of just randomly orientated in my ice. So I have, all orientations in my ice. And then when I image them, it's just like when you look at an object, right? You only see one 2D view. So we see there the top, the bottom, the side, depending on the random way it was frozen. And then we use computer algorithms to reconstruct that. So the computer takes in and it estimates, okay, well, I think this image was from the top. So now we know what the top should look like. And this image was from the side. So now we kind of know what the side looks like. And it does that for all the different images and all the orientations until it has a full picture of what the whole shape should look like. I don't know if any of Miri or Jonesy just went away, have anything to add on the orientation side of things.
Yeah, I can say that everything you said was correct. And you can kind of think about it, like if you go into nature and you take a picture of a bird, you can't always ask it to be oriented the way you would like it to be oriented. And let's say you want to build a 3D model of the bird, you'll have to collect many pictures of the bird so that you see it from all directions. And then perhaps change the sample preparation. So the way that if you want to get a different picture of the bird, maybe instead of sitting under the tree, you'll sit like at the window and then you'll be eye level with the bird. You can kind of change the sample preparation, the timing that you take for freezing, the salt concentrations, as other people mentioned, the ice thickness, and kind of tiny tricks that we've learned to do to kind of try and help orient molecules in different ways. But yeah, it's sometimes a big problem for molecules that are asymmetrical and have a preferred orientation. So we kind of always see them kind of like this, but maybe we're interested to see what's going on this side.
Yeah, so just to add to that, I'm incredibly lucky that I deal with things that are very small and often globular, so they don't have a preferred orientation, right? So what Miri is talking about is, imagine, right, you have something that's very pointy and it prefers to be pointed up instead of sideways, right? So I'm lucky in my case, for my samples, I don't have that, but many people doing Cryo-Em often have that problem. So that was great to add, Miri.
Okay. Let's pursue that a little. So you showed these pictures that your British colleagues took of the RNA actually embedded in a ribosome.
Yeah.
So when you use a big structure like that, you get presumably fewer images and more oriented. Could you just describe the relative difficulty of seeing the RNA in situ and free in a solution?
Yeah. So if we're comparing, imaging a ribosome and imaging a tiny little RNA, there's a lot of factors that are different. So one is the preferred orientation, right? The larger it is and the more asymmetrical it is, the more likely it's going to prefer to be one way or the other. The other factor is how easy it is to know what your view is. So if you have something very small and noisy, it's going to be hard to figure out whether it's a top, a side, or a bottom view. And that's the problem we have with small RNAs, is actually figuring out how is that particle oriented, right? Whereas something as big as the ribosome, it's often much easier to see and figure out if it's a top, side, bottom view. So we can much more easily correctly assign the view when we're reconstructing the 3D shape for something larger. So that's the second factor. So we have, We may have more preferred orientation, but it's much easier to guess what the orientation for each image is.
Anyone else want to comment on this? Okay, let's move on to a different subject now. Anthony would like to know how big a frame shift can happen. Is it really just you shift by 1 or 2 base pairs, or can you have larger frame shifts?
That's an interesting question. So all the ones I know of are either -1, so move back 1, or +1, so move forward 1. I don't know if Jonesy or Miri has heard of other scenarios of frame shifting in nature.
Guess not.
That may be all that we know about. And as with biology, we may simply not have discovered it yet, right? So I'm not saying it's impossible, but we just don't know about any yet.
I must say that this idea of a frame shift was really novel and mysterious to me. And Gordon had a question. I'm going to read what he said and then interpret it a little. He said, it wasn't clear to me if the slippage is necessary in viral protein production or whether it is an exploitable defect. And maybe you should comment on that, but there's a more general question, which is, why should there be frame shifts? I mean, in human DNA, the proteins are laid out with all this like dark matter between them. Why does the virus form itself in this way? Presumably it has some evolutionary advantage. What is that?
Okay, so I can give a shot at this, and I think Mary may also be able to give a shot at it. So viruses are quite different from humans, right? They only have like 30,000 bases. That's their whole genome, right? Humans, billions of bases, okay? So we're dealing with very small scale. And the reason they're so small is they have to package the full genome into this little virus that then has to travel around and spread from human to human, et cetera, right? So by having one part of the RNA sequence code for 2 proteins, they're able to code for more proteins in a smaller genome, which in theory can help them propagate more. Now, as I think Gordon said in his question, that makes it exploitable, right? It's something that if we mess with it, if we make it slip too frequently or not enough, now the viral proteins are all made in different concentrations. We have different amounts of viral proteins, and the virus just doesn't function as well anymore. So that's kind of our aim in learning more about how frame shifting works, to try to stop it and use it to harm the viruses. Yeah.
Hey, Miri, you want to add something to that?
Everything that Rachel said, and then same, when you have a frame shift, the virus can exploit it to make different proteins at different ratios. For example, there might be a protein that it needs a lot of and some other protein that it needs only a few. So it can make the frame shift less frequent, and then you can play with different ratios and build different structures and different machinery that is required for the virus to survive. And then the other point is, as Rachel said, the viral genomes are very small compared to, for example, human genomes. And I study HIV, which has an even smaller genome than the SARS-CoV-2. It has only 9,000 bases. And this way it's able to use just one mRNA that it's making, and then it can frame shift when needed.
So HIV really uses this mechanism of producing a lot of something and a little of something else by frame shifting.
Yes, the ratio is very important for HIV.
Other comments on this question? Jonesy, you want to weigh in? No?
Nope, I agree with Rachel and Miri. They're spot on with that. So.
Okay. There's one more set of questions. Oh, I'm sorry. Go ahead.
I kind of want to refer to an earlier question by Anthony. He asked, how big can a frame shift be? I want to mention that there is also other kinds of events that are not a frame shift that RNA can cause. And those can be much larger. They could sometimes be like 100 bases long. For example, in the process where RNA is being copied to DNA in the HIV virus, there are events of template switching where in HIV case, the enzyme, not the ribosome, a different enzyme called reverse transcriptase, can jump very long distances from the site that it was into a different site. And also in SARS-CoV-2, there's also events of template switching where the enzyme that's copying the RNA is actually jumping and sometimes 100, sometimes 1,000 bases difference.
But actually, I've got to ask you, are those 1,000 bases or 100 bases along the chain, but maybe they're near to each other in space, and you could learn about that with this method?
That's a great question. And that's exactly what I'm looking into, and also other people that are studying this template switching event. We're looking into how the RNA structure can regulate this kind of transfers that when you look only at the 2D sequence, it may look very far away. But when everything is folded, it kind of brings those areas right next to each other. So this kind of 3D moving puzzle can come together and aid the enzyme to transfer from this spot where it was into the new spot.
Very cool.
So I think I just want to add to that. So you know how we were looking at this, it was only 80 bases roughly. So we could see the 3D structure of that. But to study the things that Mary is talking about right now, what we would want is to be able to image the full genome, right? That would be the ideal. Right now, our technology is not there right now. We can only, image things that are compact and fold up. But that would be what we want to get to, is really being able to see how the full genome looks like in 3D space, and maybe these regions are in fact close together. So hopefully Cryo-Em will be our route to that path, and we'll see.
Okay, very interesting. There's one more issue that's in the questions, so let me ask you about that. With proteins, we've been imaging proteins for 70 years with X-ray structures. And so there's a lot of experience, a lot of data, and now there are new computer algorithms, like one called AlphaFold, that are pretty good at predicting the structure from the sequence. Where are we in that with RNAs? Is it intrinsically more difficult? And when are we going to get there where we can just take the sequence and by computation instead of by experiment, learn about its structure?
I can take a stab at
Go
that.
for it.
So I would first start and say that there are a couple of programs out there that just based off of like thermodynamics and minimum energies are able to actually fold RNA and generate predictive tertiary structural models. One is like Rna Composer, for example, and this is something that you can just take a secondary or a primary sequence and even details about the secondary structure from like different various experiments that may be carried out in a lab and generate a tertiary structure. I think what's interesting about AlphaFold, and this was something that Rachael mentioned in her talk, there are about 160,000 protein structures that exist for proteins, and for RNA, there's only about 1,600. And AlphaFold was generated because we have so much data, about different proteins that we're able to actually, generate these more accurate models and develop models to basically get a sense of what protein looks like. And I think in the absence of data, because we don't have so much data about RNA, it's more difficult, or we're just not quite there yet. I wouldn't actually even say it's difficult. If you don't have data, it's kind of hard to actually like generate a model. But I think with more time and with more approaches where we're able to actually experimentally determine structures of RNA, we're actually able to build that library or build that database of structures to develop something that's more similar to AlphaFold to improve the accuracy of modeling tertiary structures, like without having to use Cryo-Em or NMR or X-ray crystallography. So I think a few more, maybe another decade, maybe not so far, but maybe 5 to 10 years or so, maybe we'll hit that, hit the AlphaFold stage with the RNA.
Interesting.
Yeah, I think that's the goal for a lot of us doing RNA structure is like, we'd be happy if we had that predictor. We just don't yet. And that's why we're trying to solve more and more structures. And I just want to add on when you said, oh, it may not be more difficult. There's a lot of people that think it's actually an easier problem. And one of the basic reasons for that is, remember how proteins have 20 building blocks? Well, RNA only has 4 building blocks, right? So actually we have a lot less possibilities of how we can build our RNA compared to when we have 20 options, right? So our search space is searching for the structure is a little smaller. Does that make it easier? We don't know yet because we don't have the data to actually try, but it could make it easier perhaps.
Okay,
Yeah.
and easier also to learn how to fight these things.
Yep.
Really understand.
Exactly. Once we know the shape, we can think of ideas of how to fight it, right? So
Exactly.
yeah.
Okay, well, wow, this has really been a fascinating discussion. Thank you all very much. We really appreciate your insights, and I hope people out there have learned a lot. We'll have another one of these SLAC Public Lectures at the end of May. That'll be on a very different subject, using quantum devices to explore the universe. And so I hope you folks will all come back then. Meanwhile, let's thank Rachael and Miri and Jonesy, and we'll see you all next time. Bye-bye.